Okay, so what we were doing now is the fact that we're looking at, this is the wrong one. That's not it either. We're looking at how hormones now are going to mobilize the fats that we have stored. So that's important. So we're not talking about fats that are from the diet necessarily, but we're talking about the mobilization of stored fats, which, like I said, is really important to know the difference there. Bring up so that way I don't overlook anything. Give you a moment to, and it's not important that you write down every single, words on some of these. And I'll put this, make it available on my part later tonight or tomorrow. If I haven't done it by sometime tomorrow, just send me an email, and that way you have all the parts of it. But all of us are just going to look for the logic, and some of it should be a play on things that you've already learned from anatomy and physiology, but you may have forgotten. But the first thing is we have epinephrine and glucagon. Okay? And we've actually talked about these already a couple of different times. We have epinephrine, which is also commonly called, what's another word for epinephrine? Adrenaline. That's your fight or flight, running from a bear. I always put a little disclaimer. If you're going to run from a bear, do you run uphill or downhill? Downhill, because with bears from A, they fall, whereas they can actually run really fast uphill. I don't know why I remember that, but I do. Just like I remember, you do not eat bear liver. Bear liver is toxic, which actually that is biochemically related. Does anyone know why bear liver is toxic? Too much vitamin A. So some of the higher level mammals and stuff, they have too much vitamin A, and so you actually have vitamin A toxicity. That's stuck with me. Okay. But we have epinephrine, we have glucagon. And glucagon, what does glucagon signal? Like, when does glucagon kick in? Not enough sugar where? In the blood, okay? So like if you've been fasting or if you're in between meals and things like that. They trigger in our GPCR, which stood for G protein coupled receptors, which we've seen, which actually I kind of like now that we did chapter 12 this semester. Usually it was done before Christmas and people would forget about it and I'd have to explain everything again. But it activates adenylyl cyclase, right? What did adenylyl cyclase do? It's ultimately going to make CAMP, okay? CAMP, cyclic AMP, triggers pKa, which pKa stood for protein kinase A, and the A, once again, was just indicative that CAMP was. Remember, we've already had some of this, I know like in chemistry of cancer, we even talk about sometimes there are different types. Remember, we had that, for chemistry of cancer, we had that one that they called pKa, whatever, where it was DNA-dependent protein kinase A. But yeah, there are multiple protein kinase. And protein kinase A, what does it do in general? What do kinases do? They're adding phosphates, and they utilize ATP or GTP. This one's ATP. Okay, so that means it's going to be phosphorylating something. It actually phosphorylates lots of different things, but with respect to what we're talking about here, is it's going to phosphorylate, an important word there is perilipin or perilipin. I'm going to say lipin. And technically it's perilipin A. If you just had written down perilipin on the quiz or exam, I'd give it to you. This is a protein. Because peri means why, if you even think about around the perimeter, like surrounding the lipids, so that tells you, gives you an idea of where it's at. Right? This is a protein that's on the surface of where the lipids have been stored in the adipocytes. So that's the important thing. We have epinephrine or glucagon. What does it make sense that those two hormones would be utilized for this entire process? Why do we need epinephrine? And to what? Fight or flight. So why does it make sense you're going to be mobilizing fats? You need that energy. You need that extra oomph. And then what about glucagon? Why does it make sense that glucagon would also do this similar process? You need the energy, right? You need to be able, sugar is low, we're going to have to do that. And that's one of the reasons why if you go on a low carbohydrate diet, you actually start to lose weight, lose some of your fat deposits because of the fact that you're going to start to utilize some of your fat deposits for energy. Okay, so what perilipin does is it's involved in activating or recruiting, bringing into action the lipases. Technically, it's called hormone-sensitive lipases, which just makes sense because there are other lipases. This is one that's just as a part of this cascade, which are lipases, and it brings them to the surface of the lipid droplet. Okay. In general, what do lipases do? They break down lipids. See? Mm-hmm. Now, sometimes I call these tags triacylglycerols, same thing as triglyceride. And so we're going to find out, this is just to review, glycerol, because it's always good to know. Glycerol has how many carbons? 3. 1, 2, 3. And what makes glycerol glycerol? There are 3 hydroxyl groups on one on each one of these. You know. And then a triglyceride, what makes it different is the fact that, and I shouldn't have drawn that all in like this way, but these hydrogens are replaced by an acyl group. I'm just going to call it R, okay? And so that's why it's a triglyceride, because then there would be 3. So instead of that, that would be a carbon with a double bond to oxygen as an R. And then same thing here. This would become a carbon, double bond to oxygen, and then an R. The R's could be the same, they could be different. And what lipases do is they're going to break these bonds down to release free fatty acids and glycerol. Okay. And these are going to go in different pathways. I think, yes, I'm going to start off with the free fatty acids. So what's going to happen here is the free fatty acids, FFA, not Future Farmers of America, which was one of the most popular clubs whenever I was in high school, since I was in a rural area.
I was in it.
I was not. I was not one of the cool kids in the FFA, I guess. Okay, but they're going to enter the blood. Now, that would be a potential problem. If we just had the free fatty acids go directly into the blood, what do you see as a potential problem here? What's the overall physical characteristic of a fatty acid? They are hydrophobic. They would clump up. That would be bad. So we have to have something there to help it out. And our good helper is, it is an emulsifier, but I mean, in of its own sense. But what one is it here? What protein in particular is going to do it? Albumin. Okay, technically it's serum albumin, which means it's in the blood. We're going to see this a lot. I like to think of it as almost like the blood's taxi cab or something, because when you take a lot of medicines, the moment it's what happens, or you can almost think of it as a sponge, it sucks up a lot of the medications, sucks up your free fatty acids, all that kind of stuff, and it just goes through your bloodstream and drops it off. So it's like mass transit. By the way, this is one of the reasons why it's one of the proteins that you have in the highest concentrations in your body. Okay, just because of the fact that you have to have lots of it, to bind pretty non-discriminately in order to take it from place to place.
This is only found in the blood, or?
Well, serum albumin is going to be found in the blood. Mm-hmm. Okay. And here, like I said, it travels through, it's going to go to the muscle, it's going to go to your kidneys, wherever you need the energy. Okay? But where does glycerol go? Remember, we talked about there are two parts in the lipases, the fatty acids, the lipids, and then we have glycerol. Where do you suppose glycerol, what metabolic pathway can we utilize glycerol? No, what metabolic pathway can we use? Because remember, we're running energy. Ultimately, your body is going to want to use every little piece that it can. It's glycolysis. Okay, it's going to enter in through glycolysis. All right, so now we have to think about what it looks like and where it can enter into glycolysis. So if we review glycolysis, in the beginning, glycolysis, traditionally, what was the very first, in step one, what did you start off with? Glucose. And how many carbons was in it? 6, right? That's why hexokinase and so on and so forth. Ultimately, you made pyruvate. And how many carbons were in pyruvate? 3. So we know, just by default, glycerol has 3 carbons. That means it's going to be in the lower half. Okay, you can eliminate all of this before then. So it's going to be in the lower half of glycolysis. Okay, so this is what we're going to be making and doing. We actually talked about this a little bit on a quiz or an exam, I think, but we can. Okay, and so we have glycerol. This is the metabolic pathway to ultimately convert it into something used for glycolysis. Okay. Give everybody a moment to get caught up. You wanted to abbreviate. And it's, yes, technically, if you notice, it's going from glycerol to L-glycerol 3-phosphate. So it is an antiomeric at that point in time, but if I won't require that you know that it's L for that point. But your body, because of the enzymes that are used, specifically will make it. Once again, we're going from something that's achiral, because see, this carbon has 2 identical things on it, to something that's chiral. Okay, so we have glycerol becoming glycerol 3-phosphate, and at this point in time, I would expect you to be able to draw that. I mean, I think that's fair to say. We've gone over glycerol so many times, and we have ATP going to ADP. So, what class or classes of enzymes could this be? Kinase, and this is one of the kinases. All right. So, if you were going to name this, what could you name this one? This is glycerol kinase. See, knowing is half the battle. G.I. Joe. I don't know, you guys. That's well before your time. But back in the 70s and 80s or whatever, whenever the original cartoon was on, they would always end with like a moral story or whatever. And they'd always tell you, knowing is half the battle. Yo, Joe. Okay. Now we have glycerol 3-phosphate becoming dihydroxyacetone phosphate, which I more commonly call DHAP. Okay. What's the only difference really between glycerol and DHAP here? Glycerol 3-phosphate and DHAP. Right. And so what kind of reaction is this? We went from an alcohol to a ketone. It's going to be a dehydrogenase because it's utilizing NAD to NADH. Is it being oxidized or reduced, going from the alcohol to the ketone? Oxidized. Right, but yes, this is a dehydrogenase. See, that's why it's important to know your enzyme families. Sorry, it doesn't come up very well to read, but it's a dehydrogenase. Maybe if I change this, it'd be fatter. If you were naming this one, what would you name it? There we go. It's glycerol 3-phosphate, which I don't know that I left enough room, but there, glycerol 3-phosphate dehydrogenase. Then we have, and have we seen DHAP before? Yes. What was some of the places? What's one of the places that we've seen DHAP? Glycolysis. And DHAP becomes glyceraldehyde 3-phosphate. Does anyone remember what enzyme does that? Triose phosphate isomerase. Yeah, this is, so we've actually technically entered in, DHAP was where we entered in, but yeah, so this is the triose phosphate. I'm going to, once again, abbreviate isomerase. Triose phosphate isomerase. And so, see, it's not nearly as difficult once you learn. I mean, we don't see epimerases very often. That's just going from one enantiomer of a sugar to another enantiomer of a sugar, same sugar. We have lyases, which is where those were involved usually in the formation or more likely the cutting of like carbon to carbon or carbon to sulfur bonds, carbon to nitrogen bonds sometimes. Some of the other ones that we don't use very often. Well, we haven't done oxygenases yet, but we will later on. Okay, but otherwise we've done a lot of these so far. Ligases. Okay. Sorry, this is just going through to show each one of these. They're pretty things. So this is a little summary. I like this little cartoon because, well, it also reminds me of Frogger, but but we have here. Obviously, this is a very simplified, and so where we have the hormone. What hormones could this be? Once again, epinephrine or glucagon. So they bind to the receptor, they initiate their GPCR, we have ATP becoming CAMP activates pKa. All right, so pKa does a lot of different things, but with respect to mobilization of fats, what it does is through perilipin, it also, first of all, which we didn't go over, it phosphorylates the lipase to help make it active, but it phosphorylates perilipin. Perilipin is what will bring in the lipase down here to that lipid droplet. Remember, this is all occurring in the adipocyte. Many times, erroneously, people just think of adipocyte, which was a common, we say adipocyte, fat cells. They just think that they're inert, that nothing is going on. But that's not true. There's lots going on because it's your energy storage, right? For long-term energy storage. So once the perilipin has activated the hormone-sensitive lipase, it takes the tag and it breaks it apart. The glycerol will ultimately undergo to glycolysis. And then the free fatty acids, which they're fine inside the adipocyte, but they would be bad out here by themselves. So then what happens is we have our little mass transit system coming through here, the serum albumin, which will bind up the fatty acids, take it to wherever it sees the fatty acid transport that needs it, and therefore it gets transported in, which will ultimately undergo beta oxidation, citric acid cycle, everything that's needed to get the ATP ultimately. We're going to find out that it gets completely broken down to carbon dioxide by the end, which of course we didn't exhale. And so, no, we haven't. Beta oxidation, we may start that today, okay, because we're about done with this part. Okay. All right, so once again, like I said, that was an oversimplification. What happens once we get to the cell that's needed? Once it gets there, it has to get into the mitochondrion, okay, which that's the technical term for a single mitochondria. Okay, so the mitochondrion, but once again, it can't go directly through. The mitochondria is really picky in that sense. It's got two membranes. It just doesn't go directly through. Okay, so that's another way you can think of another way of doing like checks and balances, so things don't get too out of whack. And I apologize for the next picture. I think it's really ugly, but it came from the old book. So since it can't pass directly through, it gets activated by CoA, which CoA is short for coenzyme A, okay, which a lot of times we also say is CoAsh, okay, which gets added on to something. And then there's a transporter that recognizes that and will take it on in. All right, so I just wanted to remind everyone that if I say acyl, or this is an acetyl group, but acyl group in general would just be wherever you bind something, and this R can be quite large. If it's coming from some of the arachidonic acid derivatives, it's got 20 carbons, so that's why it can be really large. And so here, what happens in this, which, let me make it smaller because part of it got cut off. What happens here is overall we take that free fatty acid, we have a carboxylate. That carboxylate cannot pass through all the membranes of the mitochondria to get where it needs to be. So first an ATP is utilized. This is where the phosphate of the ATP is not added on. It's the AMP, and it's for the same reason. AMP is an excellent leaving group. You can imagine something coming through a nucleophile, smacking to that carbon, boom, sound effects optional, making a tetrahedral intermediate, collapse, kicking that off. Okay, so that's why AMP is a great leaving group. So that's why it's called an acyl. That's generic. Adenylate intermediate. And it's all one enzyme. Then acetyl-CoA comes in. I'm not acetyl-CoA. Coenzyme A comes in. The sulfur is technically the nucleophile that will do the smacking. You get a thioester. And once again, this could be quite large. In fact, it usually is quite large. And then AMP, which can then be regenerated later on to make ATP. So that's why overall they're showing you carboxylate, ATP, coenzyme A, becoming a thioester, AMP, and this inorganic pyrophosphate.
So this is one of those fatty acids.
This is the one of the three, because technically for every one that got broken down to be 3 fatty acids, all 3 fatty acids undergo this.
And what broke the fatty acid out of the carboxylate?
No, the fatty acids are lipases.
Okay.
And so what it would look like is when the lipase is done, it's going to look like this. Because that's what it does. It will utilize, like, essentially it's a hydrolysis. So water comes in, smacks the carbon, the glycerol, and pops off the carboxylate. Well, technically it smacks the carbon of the carboxylate and pops that off and then leaves the alcohol from the glycerol. But yeah, that's where it comes from. So what class or classes of enzymes can this be? You realize it's an ATP. So what classes have we seen so far? You can have a kinase, which kinases, what do they do? They remove, they do transfer of phosphates. What other class of enzymes? Synthetase, and this one's a synthetase. Now it's the tricky part. If you were going to name this synthetase, what would you name it? What's one thing, I mean, obviously we're naming it after this. So what's one part on there? Oh, there's an acyl. Awesome, that's usually not the part that people get. Okay, there's an acyl and a CoA. That's it, acyl-CoA synthetase. Because the CoA means it has to have that sulfur already on it, and so that's why. And a lot of times people erroneously will say acetyl, and I even say it when I talk a lot of times, because most of the time we talk about acetyl groups, but this is acyl because it can take a wide range. It's more promiscuous in the end. It can take everything from 12 carbons to 20 carbons. So it's acyl-CoA synthetase.
Dr. DeSarts, you had said a couple lectures ago, chylomicrons, right?
Mm-hmm.
Is that the same thing as fatty acid? What was?
No, the chylomicrons are what that are used in the transport to get the cholesterol esters and the triglycerides for storage.
Okay.
Mm-hmm. Yep. In your blood, rather than, because serum albumin is doing the free fatty acids, but it doesn't do the triglycerides and everything like that. It would be too bulky. Okay. So there's that. Now, here we go. This is our good friend, the mitochondria and the cytosol. So we have, I just want to point this out, we have the cytosol. Remember, we couldn't get the free fatty acid in through, the first checkpoint. And that's probably not good. It's almost like a bouncer at the at a club, okay? And so we had to first. That is not faith integration. I can't think of anything else. But if it helps you remember. Right. So we made it into the acyl-CoA. Then we have this transporter that will recognize that and to take it to the intermembrane space. So we have this intermembrane space. But there's another checkpoint here. Whoa, that was big. This, didn't mean to do that. This does not go directly in. So, your body has all these checks and balances just to make sure it's just not willy-nilly and that, there aren't fetal cycles. And that's why there are a lot of metabolic disorders where maybe it targets one point or another, and sometimes there are redundancies. But what happens here is we have another transporter that's going to come on. It's called an acyl-carnitine transporter. It's a mouthful. But I want to get that off of there. There we go. All right. So These transporters recognize another, well, technically it's amino acid, but because it's got an amine and a carboxylic acid, but it's not the kind of amino acid that we've seen for proteins, okay? It's called carnitine, which some people may have heard of carnitine before. What is sometimes carnitine used for? What do some people utilize the carnitine derivatives for? For utilizing, for energy and stuff like that. Maybe it's a supplement. This is what carnitine does. Carnitine actually is going to go ahead, and it's got a free carboxylic acid on it as well, and carboxylate, I should say. And it's going to displace the coenzyme A. So that way the coenzyme A can then go back out here to do its job again. And it becomes what's called an acylcarnitine compound, which then goes to that same transporter. And then we have the same enzyme, which is, notice this enzyme is located inside the membrane, the inner membrane, to go between the matrix and the intermembrane space.
Question.
Yes.
One, do we have to know how to draw carnitine and acylcarnitine?
No.
Okay, and
Well, I mean, I would expect you, if I give you carnitine, that you'd be able to put the R carbonyl group on it.
okay. And also another question: how does coenzyme A get out and back into the cytosol?
So that's that see, that's why I don't like this. Like if I was doing it, there's a transporter there and a transporter here. And so it can then pass through the outer membrane and it goes back out. And then likewise, we have a different set of transporters that's going to recognize and bind the acylcarnitine to take it in and out.
So the acyl group lets it run off as carnitine.
Pardon?
What about the acylcarnitine lets it come in?
This is a transporter. I'm just not very good at drawing it. And it's called an acylcarnitine transporter. So it recognizes that carnitine group essentially to allow it to go back and forth. So carnitine is just shuttling it. What enzyme do you suppose this could be? This enzyme right here. It's the same enzyme. I don't particularly like it. Wait, I think someone's saying, what is it? It's an acyltransferase because it's literally transferring the acyl group from one thing, here, coenzyme A, to carnitine. Okay, so it is. This one in particular, I'm running out of places to write. This is starting to look like a really bad sports.
I'm just a little confused. So the acyl. What would be the name of that acyl-CoA? That's not acyl-CoA. The RC doesn't belong.
Here, wait a minute, let me see.
What would you name that? What's coming into the intermembrane space?
Let me see 2 times.
Sorry.
Here we go. Okay, so now it's got my drawings off of it, so that way we can hopefully see. So what was your question now?
So
Oh, which actually you see the answer to the thing as well.
what is the name of what comes into the intermembrane space from the cytosol? So after it gets acylated, what is that called? Like, what is the name of that?
This is still acyl-CoA. It's exactly the same thing. They just didn't write the name down.
Okay, so acyl-CoA.
Acyl-CoA goes through, and then this enzyme, which is called carnitine acyltransferase, transfers the acyl group to carnitine to make acylcarnitine. The coenzyme A then goes back out to repeat the process.
So carnitine is already there.
You have a pool of carnitine here. Carnitine can go. What this means is it can go back and forth using that transporter. Okay, so can acylcarnitine, but carnitine acyltransferase is this enzyme that literally transfers that acyl group from the coenzyme A to the carnitine, so that way it can bring it in here, and then it does the reverse. Okay, and this is how we get that what had been the free fatty acid from the outside to all the way to the matrix, which is where beta oxidation occurs. Remember, it's always important to know where the metabolic pathways occur. Glycolysis was occurring out here. Gluconeogenesis was one that occurred multiple, depending on where you started, because go all the way through. Beta oxidation occurs inside the matrix. It's highly, highly localized. Where did the citric acid cycle occur?
In the inside the balloon. inside the mitochondrion.
Okay, what was that called?
Matrix.
The matrix right here. So it's also occurring down here as well. Okay? So that's why effectively we were able to get the acyl group, which is where is need for beta oxidation, from all the way from outside. Actually, we now talked all the way from the fat cell, where it was stored in a droplet, to the mitochondrial matrix of whatever cell needs the energy.
So pretty much just that acyl group is what's coming in, and the coash is already there. So it's just reading.
Yes, and so this is actually, you're actually bringing up something that's really good. We only have the coash, the coenzyme A, we have a limited pool of it. Just like we have a limited amount of NAD and ADH, we have a limited amount of acetyl-CoA. I'm sorry, see I did it again, of coenzyme A. And there's one pool, one section of it, or one population of it, that has to stay on this side, either in the intramembrane space or the cytosol. There's another population of coenzyme A that's inside the matrix. It doesn't go back and forth.
How did it get there?
So you have to have it synthesized. Okay, there's a small pool of it that's going to be synthesized effectively. Same thing, just like we're going to find out is NADH doesn't get to just directly go back and forth. Okay. All right, so that's the end of this section.